All right, great. So I'm currently sharing my screen. Hopefully everything is going all right. Thank you for this great opportunity to share our story with you guys. So our lab, I'm from Spelman College, and our lab is interested in how seed develops in general. So in this slide, we're seeing corn kernel on the left and an Arabidopsis seeds on the right. So the basic composition of a seed in angiosperm is that the outside layer is the seed coat. This is a diploid structure. And then the embryo, this is one of the double fertilization product, the diploid as well. And then in between the seed coat is the endosperm. This is a triploid structure. So the endosperm can be further divided into three spatial domains. The region that's closer to the embryo is called a micropylar endosperm. This is a region that's believed to be interacting with the developing embryo. And then on the opposite end is the chalazal endosperm. This is a region that's closer to the maternal vasculature. And in between these two domains is called the peripheral endosperm. So our central questions are, how does endosperm grow and differentiate in the three distinct domains? And what are the effects of the endosperm on the developing neighboring organs? So initially, this is a phenomenon that piqued our interest. So in this slide, you're looking at a series of cleared seeds. Initially, the endosperm develops as a syncytium. So this is the nuclei are dividing without cytokinesis. So this period called the syncytial stage lasts for roughly 3 days. And then somewhere here between 3 and 4 days, the endosperm cellularizes and then the embryo growth takes off. So as you can see here, in a period of 3 days, this is how much the embryo has grown during the syncytial phase. And then from 4 to 7, this is how much the embryo has grown also during 3 days during the cellularized stage. But you can see that the embryo growth rate is a lot faster after endosperm cellularization. So we decided to approach this problem from the point of nutrient supply. So seed development relies on the maternal plant for the support of nutrient. The carbohydrate comes in through the vascular structure called the phloem in the form of sucrose. This is a disaccharide. So it's deposited right here in the chalazal region. So the sucrose comes out of the phloem and it's converted by an enzyme called invertase to glucose and fructose. So this reaction decreases the sucrose concentration in the apoplastic space, generating a sucrose gradient. So higher invertase activity is in general correlated with a faster delivery and faster seed growth. So this is what's happening on the chalazal interface between the maternal plant and the endosperm. So on the micropylar interface between the endosperm and the embryo, much less is known. So we don't really know how the nutrients are delivered from the endosperm to the embryo. So what we initially did is to look at this at whether they're invertase or invertase inhibitors or sucrose transporters. Any of those are specifically expressed in this region called the micropylar endosperm. So to make the story short, what we have discovered, this is what we published in 2018, are two putative invertase inhibitors. So here you're looking at promoter GFP fusion. So you can see that those two genes are both expressed during the syncytial phase in the endosperm, and then their expression is higher in the micropylar region. So our hypothesis is basically before cellularization, the invertase inhibitor turn off the invertase and then shuts down the flow of nutrients. And then after fertilization, the invertase inhibitors are turned off, and then that release this flow, and then this is correlated with faster embryo growth. So to test this hypothesis, we upregulated invertase inhibitor in the cellularized endosperm. So we used the promoter to topically express invertase inhibitor in the micropylar endosperm, but at a later stage. And what we see is delayed embryo growth. So sort of supporting our hypothesis. Our next question is, what are the upstream regulators that turn on the invertase inhibitor? So the transition from the syncytial phase to the cellularized phase requires a chromatin remodeling complex called the PRC2 complex. One of those genes is called FIS2. So as you can see here, when FIS2 is in FIS2 mutant, the embryo growth rate is a lot slower comparing to the wild type after 4 days. So our hypothesis here would be, does this phenomenon involves invertase inhibitor? And actually it does. When we measured invertase inhibitor messenger RNA level using QRT-PCR, we see that in the wild type, the expression drops after endosperm cellularization from 3 day to 5 day. This is the normal pattern in the wild type. But in FIS mutant, we see upregulation of the invertase inhibitor. So this basically means that this complex is involved in turning off the invertase inhibitor after cellularization. So, and then what are we're also interested, are there any transcription factors in this regulatory pathway? So this is the work that I did when I was a postdoc at Ramin's lab at U of Arizona, that this is a group of type 1 mass box genes that are expressed in the syncytial endosperm. So as you can see here, this is also promoter GFP fusion for those transcription factors. So their expression pattern are very similar to invertase inhibitors. They're expressed in the same region, and then they're also upregulated in FIS mutant. So, so we want to test whether those transcription factors directly activate invertase inhibitor. So we used a protoplast transient expression system. So we isolated protoplast plant cells from the leaf, and then we transfected those cells with constructs that basically delivers the type 1 mass box transcription factors and also a promoter reporter into those protoplasts. So there are data already showing that those type 1 mass box genes form dimers. So we introduced those transcription factors into the protoplast as dimers. So in this set of data, you can see that when we introduce AGL62 and 37 as a dimer, we see upregulation of the promoter activity. So when we introduce them as monomers, right over here, there's very little activity. So AGL, the type 1 mass box genes, act as transcription factors as a dimer. Oh, I'm at 7-minute mark. Okay, so I'm going to fast forward the slides. So, so to make the story short, so we tested lots of AGL dimers. They all activated the promoter. So as I mentioned earlier, those AGLs form dimers. This is a protein interaction network. So we can see that AGL62 sits in the center of this interacting network. So AGL62 happens to have a mutant phenotype that the endosperm fails to cellularize. So we introduced the invertase inhibitor reporter line into the AGL62 mutant. So what we see is lack of expression in the mutant background. So which means that the AGL is indeed required for the regulation. So, and we also did promoter deletion assay, which identify the region of the promoter that we call the enhancer 2, that's basically required for promoter activity. When we delete this region, we see a sharp decrease in the promoter activity in the protoplast. So we also verified this in planta. So here, this is the full-length promoter activity in 8 independent transgenic lines. And then this is the D5 fragment promoter activity, very similar to the full length. And then we delete that region, we see a sharp drop in promoter activity. So we also identify cis elements in this region, that they are 3 cis element region called the CRG sites that are required for promoter activity. When we delete region around those elements, we see a decrease in promoter activity, and the effect seems to be additive. All right, so coming back to our central hypothesis. So this is what we have figured out so far. We have the PRC2 complex sitting on the top, which turn off a group of transcription factors called the AGLs. Those serve as the activators for invertase activity. So when this happens in the endosperm, it somehow influences embryo development. And we think this is accomplished by regulating the rate of sucrose transport. So finally, okay, I'm going to skip my future direction just to acknowledge the people who did the work, my fabulous technician, postdoc, and there are a number of very talented undergraduate students who are involved in this project. So the financial support, this is from NSF iOS cluster. All right, so I think I managed to finish in 10 minutes. Okay.
You did excellent. Thank you so much for that. So everyone, cheers, a round of applause. Thank you. Going first is always the hardest as well. So thank you for doing that. After we hear for our next 2 lightning round presentations, we will open breakout rooms so that you can go and chat with Dongfang. So thank you again for getting us kickstarted this morning. Up next, we will have Karmella, and I see you are with us, Karmella. So if you would like to, good morning. We'll make sure that we can get you unmuted and get your slides up.
Declutter. Okay. All right. So let me share. Okay.
Yeah, well, you might want to, when you get it in presenter mode, there you go. I think.
Yeah, I need to close out a couple of other programs because my Zoom and PowerPoint running at the same time destabilizes my computer for some reason sometimes. So, okay. All right. So let me go to presenter mode. Okay.
They look great. Thanks, Karmella.
Thanks. All right, so thanks again to the organizers for inviting me. So I'm going to just give a quick overview of, well, I'm actually a biologist who's always sort of, well, I started off as a mathematician actually in high school and college, and then was fascinated with genetics, but then sort of admired, you know, sort of like the deeper, you know, it's quantitative biology from afar. And I've always gotten involved and sought out ways to incorporate that into my own research. Whenever I had the opportunity. So I'm going to talk to you about one of the projects my lab at Emory University School of Medicine is working on right now. And as I present this information to you, I'll also contextualize by telling you a little bit more about my background and research interests. So I just want to introduce everyone to my fantastic lab. Most of these folks, except for 2, actually joined my lab during the pandemic. So I hit a growth spurt. Also a bit of background, I was at Arizona State University for about 6 years. And then sought, you know, better career opportunities. And so Emory recruited me. I basically started from scratch, graduated on my ASU students, and then, you know, started with 2 research techs, and then postdoc, 2 grad students and 2 undergrads joined the lab shortly after. So I just wanted to give a quick acknowledgement to my team. So our lab, our disease focus, so we're in the program, our biomedical engineering program, which is joint between Georgia Tech and the Emory School of Medicine. And then so our research is a very, you know, sort of disease focused. So our lab focuses on triple negative breast cancer. So I want to give you just a quick overview of that for those of you who are not super familiar with this particular disease. So it's called triple negative because the cells lack 3 key receptors that would otherwise make the cells responsive to hormone therapy. So when you're diagnosed with triple negative, unfortunately, first line of treatment is chemotherapy. And then even then, there are cases where, you know, patients who are treated, you know, the tumors reappear in other places, they metastasize about 5 or 10 years later. And then those tumors typically don't respond to chemotherapy at all. So there's definitely a strong need for, you know, more creative ways of thinking about therapy. So a bit about what we know, I guess sort of the key things that we know about the phenotypes of the cells. They're very mobile. So what happens is that, you know, in the original tumor, you'll have a lot of these cells with a more epithelial phenotype. And then there's a change in gene expression, which switches their phenotype to this mesenchymal or mesenchymal, whatever you prefer, state that has the same characteristics as stem cells. So they proliferate a little faster. They can survive, you know, sort of away from their original site, away from their original tumor, and then travel up to other places in the body and then establish other tumors. So another reason why, besides, just the technical challenge of this, right, the challenge that this disease presents for treatment, I'm also very interested in this disease because it disproportionately affects African-American women. So there's a lot of research out there really trying to pin down, oh, is it biological? Is it, socioeconomic? And I think that, what's important to me is that, even taking all those complications into consideration, in a group that is traditionally underserved medically, you also have a problem that seems to need, sort of a more creative, out of the box, more advanced, technology. And in terms of different populations, we're traditionally underserved in that regard. So I think this is a great opportunity to make a biomedical impact with synthetic and systems biology. Okay. So then just a really quick overview. I think I like to show this slide just to kind of put things in perspective. And it also is kind of a review of what I've learned, having only sort of thought about cancer for maybe 5 years. So I consider myself a continuing student of cancer. So overall, based on what I've seen, traditionally cancer treatments, there's this concept that appears over and over again, right? When you hear talks from MDs and drug developers, this Achilles heel concept. So basically we're looking for some factor that is overexpressed in cancer, right? Oncogenes, some mutant protein, and then that becomes a drug target, right? So the idea is that the cell becomes addicted to some mutant product or some, genetic state. And then if you disable it, then you kill the cancer cell. And then so I guess sort of newer, right? So an idea that emerged, after, folks discovered that you could clone genes and transfer them, from cell to cell. This idea of gene therapy got a lot of folks excited. So another thing that we know about cancer is that in the post-genome era, when we're able to actually sequence genomes and find mutations in genes. Oh, my video froze. Can you also hear me? Okay.
Yep, we can hear you good.
All right. So then, so in the post-genome era, we've, identified genes that if they were wild type, they would, sort of prevent the cancer cells from behaving like cancer. And, I may have, hold on for a second. Did my slide advance?
Can you try it again?
No, it did not.
I knew this was going to happen. So my computer froze and I'm going to have to reboot it. I don't know what it is. It's PowerPoint and Zoom at the same time.
It might also help bandwidth with turning off your camera. That helps.
Well, the problem is that I'm stuck in presenter mode.
Okay, well, here's what I can do. I'm going to go ahead and take that away from you for a quick second. And I will take that away. There you go. So that.
No, my computer's frozen.
Okay, well, why don't we do this? Why don't you want to jump out and then come back really quickly?
Yeah, I can. I'll have to come in on my laptop.
Okay.
It's gonna take another 5 minutes for the. I don't know why this keeps happening. Oh,
That
well,
is.
I got a PC.
That is okay, Karmella. We'll what we can do to give you a couple minutes, we can actually, if you like, we can move on to Janet.
Okay.
You got it. While you come back and then you can wrap back up for us. Does that sound okay?
Yeah, if you could give me it'll only take me 3 minutes to finish this.
No problem, Karmella. Thank you. The joys of working virtually. This is why we say you have to we're all in it together, right? We're all in it together. So Karmella, we'll see you back here in just a couple minutes. With permission from the group, and Jan, I kind of leave it up to you as well. Do you want to wait a minute while Karmella comes back and finishes, or do you want to go ahead?
I'm happy to do it either way, whatever you think is best.
Okay.
And Jen, while we hear it, shall I ask, would everything be okay with your sharing, or?
It should be all right, yes.
Okay.
I have it set up.
Okay, so let's go ahead and try. We'll let you go ahead and go, and then we'll come back and hear the rest of Karmella's, a quick update and the rest of Karmella's when she comes back.
All right, sounds good. Okay, that looks good on my end. I hope you guys see this as well.
We do. We want to get you and present. Yeah, it looks great.
All right, perfect. I'll look at my time. This is the shortest talk I've ever given, so I'll do my very best to stay on time. My name is Spille. I'm a relatively new assistant professor at the University of Illinois, Chicago in the Department of Physics, but we do a lot of biological work as well. So when I was asked, am I a modeler or am I a biologist, I didn't really know what to say. What we're interested in is understanding the organization of the cell nucleus, and we take a single molecule approach towards doing that. That means we do single molecule imaging to try to understand how transcription regulation works. And if I say that we do single molecule imaging, that is really only true as of yesterday. Our microscope finally arrived. There is also a national shortage of microscope technicians, but they were finally able to install the instrument that is going to do all the experiments that me and my grad students have been talking about yesterday and today. All right, so let's jump right in. The system that we're interested in is the cell nucleus. And what's really fascinating to me as a physicist is that you can pack these almost 4 gigabytes of information encoded in DNA into this tiny little cell nucleus. And then somehow in that is encoded a program that allows the cell to assume all sorts of different fates. It can be a blood cell, it can be a neuron, things can go wrong and it can become a cancer cell. So in the human cells, we have about 20,000 genes encoded in this DNA. About 5,000 of them are active, and that's what makes a cell a certain cell type. And that is regulated by on the order of a few 10,000, some people say 100,000 enhancer regions that control the expression of these genes. And just to give you a ballpark number, and let me turn on my laser pointer here. In the cell nucleus, we also have about 100,000 copies of RNA polymerase II, the enzyme that transcribes the genetic information into usable mRNA copies. So that leaves us with a lot of problems, right? So we have not just RNA pol II, we also have dozens of other factors that all need to come together on a gene promoter and an enhancer that can be located anywhere upstream or downstream from the gene to drive expression of this gene down here. So all these different factors somehow need to find the target inside of this crowded environment of the cell. So how can we make that happen? Well, our approach is experimental. So what I did as a postdoc is engineer cell lines where we fluorescently labeled some of these proteins, namely RNA pol II and the mediator complex that connects the enhancer and promoter regions. And then when we do super resolution imaging or light sheet imaging, what we found is that both of these factors accumulate in strong local foci, where we have high local concentrations of both of those factors. And we see a number of those in every single little cell. So this reminded us of a model that was proposed around the same time, a phase separation model for transcriptional control. So basically the idea is that all these proteins in the cell nucleus behave somewhat like a liquid. Most of them can diffuse around relatively freely, but they may have affinities between them or self-affinities that allow them to do something like oil and vinegar, namely form little microenvironments through a phase separation process that make this liquid in the nucleus less homogeneous and create these little microenvironments that can have function. And sure enough, when you purify some of these proteins, for example, the mediator complex or a subunit of it, or RNA pol II, and reconstitute that protein in a test tube, you do get these little droplets that look exactly like these liquid droplets that vinegar forms in oil in your salad dressing. And when you mix the two of them, then they go to the same place. So they actually coalesce into the same droplets in a test tube. So this looked very much like what we saw when we looked at these proteins in the cell. So we tried to test a a few of the key characteristics that you would expect from a liquid-like system in the cell. These are really droplets. One thing that we did, and I hope this video plays on your screens as well, we did time-lapse imaging of these live cells and tracked individual of these foci over time. And sure enough, when they contact each other, they can fuse into a single entity. And if the video did not play, here are the stills, right? So we're looking at these two condensates, they diffuse around, and at some point they touch and then they stay together, they never fall apart again. And this is full 3D data, they're not just on top of each other, we track the intensities, so these entities can fuse like you would expect for liquid-like droplets. Now this video that I showed you ran for something like 10 minutes, 20 minutes. So these are very stable entities. How can a liquid be stable? Well, that's one of the big questions. But that's something that we would expect if the constituents self-aggregate into these droplet-like structures. The other thing that we wanted to know is, well, if they are stable, are they really liquid-like, or are they more like, should we think about them like a crystal or something like that, like a more solid aggregate? So we did fluorescent recovery after photo-bleaching experiments, where we ablated the fluorescence of one of these foci with a strong laser pulse. And then we have a control cluster over here. And then we tracked the recovery of the signal over time. And now note the time scale here. This is seconds. So within a few seconds, the signal comes back after we bleach it. So on the one hand, these entities are stable for tens of minutes. On the other hand, the constituents turn over on the 10-second time scale. And that is true for about 60 to 90% of the protein fraction, depending on which of the proteins we look at. So again, that is something that is very consistent with this phase separation model. However, there's a problem with this analogy. Right? If you mix your own salad dressing, you let it sit for a while, then you know that it will separate. And that is something that is not good for the cell. Right? If all my proteins are in one place, I only get these two faces here, then I lose the ability to selectively control the expression of all those thousands of genes that I need to regulate. So maybe this is not maybe it's a good starting point to think about as a model, but for sure it is not going to be a perfect explanation for what we observe. So what we're exploring right now is if something like this is maybe more appropriate, where we have a scaffold structure like this cobweb, and then we get condensation in certain places where, for example, we have intersections of the scaffold or a high local density of nucleation sites. So this is what some of my students are working on and are going to work on with our new microscope. One thing that we have been able to do already is, for example, super resolution imaging of epigenetic marks that tell us where to find these active enhancers in the cell nucleus. And then we find these tiny little clusters. So we can use drug treatments that inhibit protein binding to these active enhancer marks. And then sure enough, we find that condensates disappear. So the question for us is, are these the nucleation sites where the condensates form and allow us to, in a spatially controlled way, create these microenvironments that can drive very efficient gene transcription? Another thing that we're wondering is this liquid model really the whole truth? If you look very closely on our previous images with standard resolution, you will see that not all of these components are always perfectly overlapping. So maybe there is substructure to these condensates that we can just not resolve with the current resolution. So again, we hope that super resolution will be able to distinguish certain organizational properties. And this is my last slide. So the last thing that we're doing is single particle tracking to really probe the material properties of these tiny little droplets inside of cells with, because standard assays that you could apply in a test tube do not apply in a cell. You can't just squish the cell and expect that everything is still the same, but single molecules can act as a probe for material properties of these condensates. So these are the folks that all joined throughout the pandemic. Ganesh, the first student, he contacted me in middle of March last year, and I said, let's meet next week. Then that turned out to be June. Nevertheless, these guys stuck with me, and we're finally gearing up to do the experiments to test these models. Right, thank you very much.
Excellent. Thank you so much, Jan. That was fantastic. So thank you. Yes, round of applause, round of applause. Excellent. So just as a reminder, we are going to finish up Karmella's, and then we're going to open up breakout rooms so that you can go and interact with all of the speakers and kind of go and ask them all of your questions. But thank you, Jan, for picking that up really quickly. That was an excellent presentation. And I do believe that Karmella is back in action. So Karmella, if you want to take kind of maybe back up one slide, just as a kind of reminder of where we are, and then awesome. Excellent. Thank you so much.
All right, so this is going to be really quickly, so I apologize for the speed. But I do want to get straight to, I guess, sort of like what we've been doing with modeling so far with our project.
So, yep, there you go. So back over where, yeah, there you go.
Okay, all right, so we sort of left off here. So we're our lab is interested in sort of a this area called epigenetic therapy, right? So this is a sort of like an emerging approach to cancer treatment. And the general idea here is to restore or enhance expression of tumor suppressor genes that have been epigenetically silenced in cancer cells, right? So rather than, rely on something like gene therapy to put, a non-mutant copy of an anti-cancer gene into tumors, which would be pretty hard because of the barriers to gene delivery, what we're doing is that we're activating genes in situ. And a lot of folks might be surprised, given all the information we're given about mutations and cancer and all these lesions. There are dozens and dozens of anti-tumor genes that are not mutated, totally wild type, but their expression has been downregulated by the cancer cell. So the goal of epigenetic therapy is to push these genes from a silenced to an active state. So far, as is typical, I guess, for drug development, because we're so familiar with small molecule drugs, right, delivering things either intravenously or through a pill. Drug development has relied a lot on identifying small molecules that can shift the epigenetic state of these tumor suppressor genes. But they've had very promising so far for blood cancer, but very limited efficacy in epithelial cancers, which I actually learned the other day through chat that 90% of patients that have any sort of cancer, it's going to be an epithelial cancer, right? So there's still a great need. Epigenetic therapy has a lot of promise, but its effect is limited in the majority of cancers. So what our lab is working on is a concept where we, instead of relying on drug inhibitors for what is essentially loss of function, right? Sort of disabling the problematic chromatin proteins that are silencing tumor suppressors, we're thinking about ways to build fusion proteins that can go and sort of scan the epigenome and land on genes that have been hyper-repressed and then force activation of those genes. So this is just some early work that I did as a postdoc in Pam Silver's lab. For the sake of time, I'm just going to point out the really exciting result over here, right? So we built different fusion proteins that included a truncated control, just to make sure that what we were looking at was an artifact. So basically what this fusion is that this is a bit of, it's like an N-terminal piece from a protein called CBX, which is a polycomb chromatin protein that can read histone H3 trimethylated at lysine 27. And then so this concept is really straightforward, but of course, with any design, you have to test it to see if it works. And then we fused a really commonly used, very strong virally derived activator to the C terminus of that domain. And then voila, it works. We tested this on a model transgene where we were able to deliberately place polycomb chromatin, right, including the methylation mark on the histone near the transcription start site of that gene. And so what we were able to demonstrate was that the gene activating function of this fusion relies on the accumulation of its target near the gene. So that was really exciting. So to wrap up, I want to talk to you about what we're interested in terms of modeling. What we observed right when we tried this on the epigenome of a cancer cell was that, going in, knowing where all of the target marks were, right, with some nice, public ChIP-seq data, what we found was that not all the genes that had the target mark responded to the fusion, And also not all of the genes that were being activated actually showed that signal, right? Actually showed the target signal near the transcription start site. So then we started wondering, okay, well, is the picture more complicated? Is our fusion sort of picky, right? Does it have a preference for certain regions? Is there a combination of histone marks, right, that poises certain genes to be activated? So lots of questions there. And so we've collaborated with a systems biologist, Chris Plaisir at ASU. And then so what he asked us for, and so I think this is a nice example of, biologists talking to folks who are doing more computational biology. He sort of, I posed the question, and then so he was able to consult us on the type of experimental data we ought to generate to really get to the bottom of those questions. So one of the things he asked for was a time course induction series. So we took a stable cell line where we could induce expression of the fusion protein and then look for genes that were either early, middle, or late responders to the fusion protein. So we suspect that our early responders are going to be like the bona fide targets of the fusion protein, whereas the late responders may be the targets of, like, let's say, a transcription factor that's being expressed from the early responder group. So right now we're investigating that. And then what we ultimately want to do is build a computational classifier where we're able to take a look at the features of the genes that are bona fide targets of the fusion protein, and then have a more, I guess, sort of a data-informed profile of those genes that are actually responsive. Okay, so then, yeah, so this is just a concept, the sort of layout of what we're thinking about, right? Taking features that are mapped, identifying genes that are bona fide targets, and then our output is, basically a profile of those genes that are responsive. And then, so we'll take that classifier and that was trained on this data set and then test it on, by using data from other cells where we introduce the same activator, look at what responds, and then basically see how good our classifier is at predicting what the targets are. Okay, so with that, I'll go ahead and acknowledge all the folks who have contributed to this project. And the classifier is actually funded by an R21 grant from NIH NCI. Thank you very much.
Excellent. Thank you, Karmella, and seamlessly picking that back up and coming back to us. So apologies for technical difficulties. Thanks for hanging in there.
No, it was on my side. Yeah, thanks for your patience.
Excellent. Thank you, everyone. So at this time, I'm going to go ahead and explain how the Q&A breakout session will work. So my colleague,